All right, it's now Wednesday. Last time I recorded one of these was Saturday and I was from home. Now you can see I'm working from my office. We're sort of having school online. Because all our labs were finished up, it's actually not that bad for my particular situation to be able to have the students do all their writing and data analysis and things like that this week. And so we're still on schedule. We're still going to have school, and we are closing out the quarter with a very vivid example of the importance of science and numbers to all of my students. And so I'd like to talk a little bit more about the numbers that have come out since Wednesday. One really important thing that I've noticed a lot of people talking about cases, confirmed cases of coronavirus. And that's a good number. The thing you've got to remember, you can't have a confirmed case unless you have a test in the first place. And because we've had problems with the rolling out of tests, at least in this country and in this state, we have a grain of salt that has to be taken with any argument based on cases. In fact, I look at cases last as far as the numbers go. I want to keep an eye on the hospitals first. This virus is bad in terms of mortality, but it's possibly worse in terms of hospital load. People who need to be in the hospital might have to be there for a long time, and there are some signs that that is definitely happening. So what I'm worried about with this virus is the potential for mortality is a huge problem, but also the potential for long hospital stays. You'll notice the NEJM example that I gave last time. That patient, 35-year-old man, 10 days or more in the hospital from this thing. This is not messing around. And so it does look like, again, this is about 10 times as serious as the regular flu in terms of severity and impact on the hospitals. So hospitals only have so much beds. We have only so many resources, and healthcare workers are human. They only have so many hours in the day. It's that's going to be the primary challenge, and that's why we need to keep ourselves healthy and socially distanced, as they say, because if we can keep this from spreading, we can not only prevent deaths, but we can also keep the hospital beds reserved for the most severe cases so that the hospitals don't get overloaded. So I want to keep an eye on the hospitals first. The problem is there's not really systematic data that comes out that I'm aware of about how the hospitals are occupied or how the people's time is being used. So you can depend on certain anecdotes, but you can't trust those on Twitter. You can depend on good reporting, like this example right here, where we actually go into Evergreen Hospital and they talk about how it's been so far, how they've handled the many cases that have come their way. So keep an eye on the hospitals. Keep an eye on the signs that healthcare is beginning to get overloaded. Deaths are another number that is very important to keep an eye on, but I want to encourage you not to look at those too closely, because when you have a small number of numbers, they can be influenced by little fluctuations one way or the other. And the fact of the matter is the deaths in Washington State are increased because the virus got into a place it never should have been allowed into, never should have been introduced into in the first place. The nursing home center that it was introduced to allowed it to spread. And so we've got to focus on preventing that from happening again, testing those cases, keeping that from happening. It was an ongoing tragedy, and it shows how bad this virus can get. But you can't just take that number by itself and then extrapolate it. Say if we've had 24 deaths over 7 days, therefore we're going to have X number of deaths over 14 days. We don't really know, and that's the thing about the tipping points and the exponential growth that we were dealing with before. So that's my advice, and keeping an eye on the numbers that I understand and I can know something about. There's another number that popped out at me. This is interesting. Even with the limited testing we've been able to do so far, there have been more than 2,000 tests run in Washington State. And I just got this off of the Washington State Department of Health website. The tests that were run, 267 of them so far have come back positive in this state. According to this, 2,175 have been negative. That means 88% of the tests that have been run, roughly 9 out of 10 of the times that we have run a test, it has come back negative. I'm not completely sure that this doesn't include also in the negative value. I think that it involves tests that actually came back negative as opposed to waiting to be confirmed. Even if it includes the waiting to be confirmed number, the large majority of people that are tested do not actually have this virus. They have some other health issue that's making them cough. And so realize that there's a lot to be learned from testing. And I'm a scientist. I want us to test more. I want all the data. One of the signs that it's really serious in the Seattle area is that it's changing the local institutions in many ways, including Dick's DriveIn. I remember when Dick's DriveIn would only take cash. You had to have money in your wallet to be able to buy a burger at Dick's DriveIn. But then I saw this. Dick's DriveIn is now actually asking us not to bring cash anymore. They switched over to allowing credit cards. I used to bring cash myself for tradition's sake. But now they're asking you not to bring cash because that is a physical medium that could spread the disease. So in Seattle, we're trying to get serious about this. Any step like this that could help at this point, let's flatten the curve, let's take a break, and let's use credit cards instead of cash if you're getting your burgers from Dick's DriveIn. Last time we looked at the viral tree, it looked like this. Now it looks like this. There's a lot more data that you can see, especially way over on the right side of the graph. So what does this data mean? Remember that most of these data, this data is encoded geographically, and so we have the red means that it's a certain distance away from Wuhan. And so all the red ones are going to be Seattle or California or British Columbia. But in fact, I wanted to highlight the ones that are Washington, as they have a tool for doing that online. If you see, I just made the Washington dots get a little bit bigger. And if I flash back and forth, you can watch the graph and you can see most of the red Washington dots are clustering in that area, sort of above my head right now. If you look closely, you can see that there is another dot far above my head. This is the third genome that I mentioned in the last video. This dot is the one that goes with the Italian genome that we mentioned before. Those are shown in yellow and green. That's how the European genomes get coded. And you'll notice that this third case has not been replicated yet. We just have one example. We don't know if it's just that one person or if they passed it to somebody else. Knowing this virus, it's almost certainly out there. We just don't know how much. But most of the Washington cases are actually branches off of what we have already talked about, the first two genomes being the first red dot all the way on the left that you can see there, leading to all these later red dots that are its sort of offspring. Now I want to look at the data in terms of letters, and again, here's the whole genome, 30,000 letters, and only a few letters out of these have been changed in each of these. We can still get the patterns by comparing the way that which letters are changed and where. So the first Washington genome had 3 letters different from a consensus sequence. That's the one that I showed you right there. The second Washington genome, if you remember, had same 3 letters different, and it had 3 new differences, and those are shown by the vertical lines on this graph. It shows it exactly where it is in the 30,000 letters of the genome and where the difference is. So you can see that there are 3 lines in the same place for the second Washington genome and 3 new lines that have developed presumably since the first genome was sequenced. I want to show you the third genome. So now I have that on the bottom, and you look, it has 6 letters that are different, 6 lines, but the lines are in different places as you look back and forth across the graph. These do not match up with the differences that are in the first genome. If you just compare them, you can't find anything that's even in the same region. But in fact, to find something that has letters that have the same changes in the same place, you have to go to the genome that I've now displayed on top. That's the Italian genome, one of the Italian genomes, and you see that that has 2 of the lines in the same place as the third Washington genome. This is why I'm fairly confident in saying that those are related because we have a whole bunch of genomes with these two letters different in Europe, it looks like it came over from Europe. We've always got to be probable. We've always got to be unsure about this. But the more data we collect with more genomes from Europe that look like this, the more it looks like this hitched a ride from Europe. And you can see that you have the two letters different. They match up and down to the third Washington genome on the bottom. So I can't help it. I'm a chemistry teacher, and so I have to have a little problem for you right now. I have a genome in the middle that shows where the differences are. And I'll give you a hint. It does not come from Washington or from Italy. So I want to ask you what you think it goes with. Does the genome in the middle fit with the genome on the top, the Italian one, or the genome on the bottom, the Washington one? If you want to take some time to think about it, you can pause the video right now and take a look up and down at the numbers. So if you had a guess, and I feel a little bit like this is one of those preschool Blues Clues type shows where you're asking the people at home to play along. If you saw that the one in the middle matched the one on the bottom in these 6 places, then you were right. Okay, these 5 places I should say. This looks like the second Washington genome, and so it fits with all those genomes. It looks like the ones that we've collected from the East Side Hospital where I showed you those pictures from previously. But notice that this was not taken from Washington. This was taken from California Cruise, the Princess Cruise liner. So it looks like the California, the one on the cruise, and the Washington version are related. Now, all we can say here is that this is a family resemblance. So what's really important to say right now, most of the Washington genomes, including most of what they've been treating over at Evergreen, are related to the first and second genomes that we found that I talked about a week ago. The California Princess Cruise genome, the one that we have so far, and we've got actually a few more as well, they all look like the Washington genomes. They don't look like the third one, they look like the first and second ones, the one that we know has been introduced and has been spreading in our community. The simplest explanation is that it could have come from Washington, but there's another explanation that's almost as simple. It's not quite as probable, but we really can't tell based on this data. Both of them could have been introduced from Asia independently. We know the first one was introduced from Asia. We collected that from the patient who was up in the Snohomish Hospital. So these both could have come from Washington, or they both could have gone through Washington. I think it's pretty likely, but I wouldn't want to, place any money on it yet. We don't know. We should collect more genomes, but it looks like this is spreading up and down the West Coast. Therefore, measures like quarantining, social isolation, canceling large services, and things like that, it appears to be warranted right now. My only thought when we're doing this is, since we've introduced these based on what the data says, let's think about what the data will need to look like for when we can go back to normal. And I want to assure you that it certainly looks like we will be able to go back to normal. But like I said before, it might take a month. So my recommendation is give me more data. Give Trevor Bedford more data. Let's get more genomes and let's get more tests out there. A test is not a genome, but it's still data. And so let's test more, let's slow the spread, and for the rest of us who aren't involved in the active work, let's take a Sabbath and support your local healthcare worker who very possibly could be overtaxed and overworked in the week or so ahead. This could go either way. You've heard the stories from Italy about how bad things are there for their hospitals being overloaded. But I also want to mention there are stories from South Korea. South Korea has been testing, like I mentioned that before, South Korea actually has been testing adequately, and their cases have gone down for several consecutive days in a row. That's good news. It looks like they can control things without complete social lockdown. However, you may have seen something that says that those cases have ticked up. When I said that they've gone down for several days in a row, that's actually old news. The new news is that they've ticked up again. This is worrisome. How worrisome is it? Well, the important thing is when you get a news report about number of cases in South Korea, you find out that the numbers have gone up. That is one data point. We have a graph where they've been testing in South Korea for weeks now. And so we need to look at the whole graph to see what this uptick looks like in context of the whole graph. This is the only graph I could find with the updated data. By the way, it's a little blurry and it's in Korean. But hopefully my Korean colleagues will be able to tell me that I've got actually this graph right. If you look at this, the blue line is cumulative cases detected. That blue line is going up as you move from left to right, as you go from day to day. The green line is for each day, how many cases did you confirm on that day? So the blue line is cumulative, the green bars are daily. And if you notice, for about a week and a half, we had that peak where it's 900 cases were confirmed. About 10 days ago, it looks like I'm counting on the fly, about 2 weeks to 10 days ago. But since then, they've actually gone down day by day. They've added to the total number, and the blue line has kept going up because that's cumulative. But each day you see that the trend has been to go down. Now there's fluctuation. As you go from day to day, you have it going up and down a little bit. When you look at the most recent bar, the bar farthest to the right, you notice that it is indeed up, but it still is the second best day since the testing peaked a couple of weeks ago. So yes, it's an uptick. Hopefully it's not the start of a trend. I would have to say that if I look at this data, it looks like the trend is still moving in the right direction. We can't say we're completely in the clear yet, but I'm much happier to see that that number is at least down from the days that were before the day before. You know, it's up from the day before, down from all the other days since the peak of the confirmed cases. So please keep in mind as you're looking at these that we're getting data points day by day, and it's important to look at the data, to not look at one particular data point, look at the data points in aggregate. South Korea still looks like they're on the road to recovery, and let's try to do what they're doing. Drive-through testing, but just testing in general. Okay, let's test as much as we can, and let's try to figure out who has it, and remember that 90% of the tests so far have come back negative. There is some hope here. We are going to get through this, and the solution is actually pretty old-fashioned right now for most of us. It's to keep clean, keep our distance, take care of our family, and take care of others, as the case may be. Keeping in mind that you don't want to be a disease vector. You want to be a disease ameliorator, a disease therapist. You know, you want to make things better, not worse. And you can make things better without even knowing it if you keep your hands clean and keep from touching your face. The fewer people we have spreading this, the better the hospitals will be able to deal with it. So again, I thought I'd talk for 5 minutes. I've talked for much more. That's the way that professors are. But the other thing I want to mention, several people have come to me with questions. I want to be clear, I am not a virologist, but I do actually work with the immune system. I'm a protein chemist who works with the immune system, so I'm kind of like adjacent to the field. And maybe that helps me explain a few things better. Take everything I say with a grain of salt. People have asked me a couple questions, and I want to answer them very quickly. Which cleaning products work best against coronavirus? The good answer is they're pretty fragile, so pretty much anything will take care of it, at least until we find out otherwise. Soap will take care of it, bleach, peroxide, basically you name it. If it's concentrated enough to take care of the average virus, it should take care of this one. I'm going to say that until I find out otherwise. Again, we need to do more tests to be sure. And then I've seen a lot of things going around social media with people saying, if I'm calling for more tests, okay, shouldn't this person be tested for the virus? I encourage you not to do that, though. Testing is something that should take place only when you have symptoms or when you have a very strong possibility of transmission. The reason is, if you test too early, then you don't have enough virus growing in your system for the test to detect. It's possible that you can test too early as well as too late. And so when the virus is multiplying enough to hurt your cells, to hurt your body, and to have the immune reaction and the cough and the other things like your body starts to react against it. That's when you need to test to see where these symptoms are coming from. Perhaps in a case where you can catch it while you're still asymptomatic, while the virus has grown up enough. But it's going to take a couple of days for the virus to even get to that level, and that's if you hit it right at the right time. So you shouldn't be asking if other people should be tested for the virus. If they're not showing symptoms, they probably shouldn't be tested for the virus yet. On the other hand, we've got all these people with symptoms. Let's get them tested, and then we'll get some data from that hopefully we can use in the future. Okay, thanks for listening. Everyone, keep washing your hands and take this as a chance to take a break and slow down and be with your family and to maybe read a book. I'd like to give credits again to nextstrain.org. None of this is possible without the site that they put up and made live. And Trevor Bedford and the others at the Fred Hutch, I'm following their lead on this. And so I can try to answer questions as best as I can, but if they say something that contradicts me, guess what? I'm going to say listen to them. All right. Hopefully I will have another one of these in a couple of days. And until then, best wishes.